Tuberculosis is a global health problem. It is one of the most important, preventable causes of death and morbidity in the world today. In the United States, the disease continues to be a significant challenge due to immunosuppressive conditions, immigration, and non-compliance with testing and treatment protocols. Mycobacterium tuberculosis, MTB, is a slow-growing intracellular organism, which makes culturing the organism slow and unreliable. Latent tuberculosis infection, LTBI, occurs when people carry Mycobacteria bacilli in their body, but the organisms are dormant and still in small numbers. These mycobacteria are controlled by the body's immune system and are still in small numbers. In this state, the MTB do not cause disease or any TB symptoms. Individuals with LTBI are not infectious. They would be negative to most TB tests, including culture and smear microscopy. Small nodules are occasionally seen on chest X-ray. Until recently, the only test capable of identifying LTBI has been the tuberculin skin test, TST. The most commonly used TST is the Mantoux test. This involves an intradermal injection of purified protein derivative, PPD, into the arm. PPD is a mixture of proteins derived from dead M. tuberculosis cultures. An immune reaction occurs within 48 to 72 hours, at which time the induration is measured. This test requires 2 patient visits. In addition, the test requires up to 72 hours to get the result, and false positives can occur in BCG-vaccinated subjects or following infection with environmental mycobacteria. The DNA of Mycobacterium tuberculosis has a region of difference called RD1 that is not found in BCG and most environmental Mycobacterium. The RD1 area of MTB encodes for 9 proteins, including ESAT6 and CFP10. These 2 proteins are used in the T-spot TB test to challenge T cells. If the T cells recognize these proteins, they secrete interferon gamma. The T-spot TB test is a simplified version of the enzyme-linked immunospot ELISpot assay technique. It identifies M. tuberculosis sensitized effector T cells activated by the presence of CFP10 and ESAT6 antigens from the RD1 region of MTB. Effector T cells are quite short-lived. Their continuing presence indicates the cellular immune response is currently encountering and fighting a pathogen somewhere in the body. Measuring the presence of effector T cells in a sample therefore diagnoses an ongoing infection. In contrast, the presence of memory T cells would only indicate that someone had been infected with a pathogen at some point in the past. Blood from the patient is centrifuged, and the number of peripheral blood mononuclear cells, PBMCs, are counted. The sample is diluted so that a standard number of PBMCs are added to each well. PBMCs are added to a pre-coated well that contains antibody to interferon gamma. Cells are incubated for 16 to 20 hours. In the presence of activated effector T cells, the TB-specific antigens ESAT6 and CFP10 stimulate the release of interferon gamma, which binds to antibodies to interferon gamma on the base of the well. Wells are washed, and a secondary antibody to interferon gamma is added. Following another incubation and washing step, the substrate is added. This produces spots on the well floor where the interferon gamma was secreted by T cells. These spots are enumerated to provide the test result. The T-spot methodology provides an effective means of identifying TB infection. The test is highly sensitive and reliable in targeted testing groups. T-spot TB provides comprehensive testing coverage for all high-risk groups, including healthcare workers, immunosuppressed individuals, TB contacts and TB suspects. The assay detects interferon gamma produced by T cells in response to RD1 specific peptides, so the test is also very specific. T-spot TB test results are not associated with immunocompromised status. This is because the T-spot TB test requires a standard number of PBMCs to be used for every patient, thereby minimizing the effects of low cell counts. The T-spot TB test is highly specific, as it does not cross-react with the BCG vaccine and most common non-tuberculosis mycobacteria. Benefits of the T-spot TB test are its efficiency, requires one single blood sample, it is fast, next day results, and it is one and done, no second visit required to read test results. Stop TB. Start with T-spot TB.